if we stick with the function that we just mentioned, forming a clot, all right, if a person, does a blood count and we find that their platelet levels are very low, that would be a bad situation to be in, because that would mean they'd have a hard time forming a clot. They'd have a hard time controlling bleeding. And, controlling bleeding or controlling blood flow in general is a process we call hemostasis. Okay, so they would have a hard time regulating hemostasis. All right, if your platelet count gets too low. If your platelet levels start to get up too high, that may indicate that a person has a clotting disorder. They may form too many blood clots, and then those blood clots may start getting stuck in the microcirculation and occluding the flow of blood to specific areas of the body. All right. So now that we've kind of just basically established what we use platelets for, let's talk about the process of hemostasis. Let's talk about what happens when we actually damage a blood vessel and we're leaking blood out and we need to control the bleeding. Okay, because bear in mind, just to review, the circulatory system is a closed system. Again, when we say that, what we mean is that blood should remain within blood vessels and within the heart. And that's, so blood, and if any fluid or plasma of the blood does leak out, it should be returned, okay, to maintain a normal fluid cycle. All right, if we are losing blood, that's not a good situation to be in. Okay, that means we're bleeding. All right, and if it's in massive amounts, again, we call that hemorrhaging. All right, then every now and then you will damage a blood vessel. I mean, you guys watching this have all cut yourself at some point in time in life. Cutting a vegetable and you accidentally clip your finger, or walking through the woods and you scrape your arm against a sharp branch, or maybe bumped in the barbed wire, or just anything, any point in time of life you hit a sharp object, you cut yourself, you notice you started to bleed, but eventually you stop. Okay, and a big part of that came from this hemostasis process. Now, before we talk about this, let's talk about the basic layout of a blood vessel. Okay, now if you look at the basic structure of a blood vessel, it comes in 3 different tissue layers that we call tunics. Okay, there's an outer tunic, there's a middle tunic, and there's an inner tunic. Okay, we call them the tunica externa, tunica media, and tunica internum, or the intima. All right, but again, we'll get more depth of this in a later chapter. But one thing to keep in mind is that these tunics of the outer tunic of a blood vessel is basically just supportive connective tissue. The middle tunic of a blood vessel is where you find the smooth muscle of the blood vessel, and then the inner tunic is where you find cells that line the inside of the vessel. All right. Now, whenever you're talking about an organ like a blood vessel that's hollow on the inside, we call the hollow area the lumen of the vessel. Okay, or if it was your esophagus, we were talking about the opening of your esophagus, we would say the lumen of the esophagus. Okay, so we could say that there are cells, within the inner portion of the blood vessel that physically come in contact with the lumen of the vessel. That means these are actually coming in contact with blood. Okay, as blood is being pumped through the vessel, blood physically contacts these. Okay, now the question you might be asking yourself is, how come platelets don't just stick to the walls of blood vessels? Because earlier I said they were sticky little cell fragments, and they start to clump together eventually to form a clot. What's preventing them from actually sticking to the walls of vessels and including blood flow? And that comes from, and the reason why they don't is because of these cells that line the inside of the vessel, and these cells are called endothelial cells. Okay, endothelial cells. Endothelial cells are very important. What they are, they're just basically simple squamous epithelial cells, and they help play a very large role in maintaining the integrity and just the overall function of the blood vessel. Okay, they may release vasoactive chemicals that can make increase the muscle tension in the vessel and make it constrict, or vice versa, relax the smooth muscle and make it dilate. Okay, so the endothelial cells are quite important. Another big role that they play is they produce these sticky chemicals called prostacyclins. Okay, and these prostacyclins, what they do is they prevent, as these sticky little platelets these clumps of platelets are circulating through the bloodstream, the prostacyclins prevent them from sticking to the walls of the vessels. Okay, they prevent them from sticking to the walls of the vessels. So that's the big role of the endothelial cells. Okay, in regards to hemostasis is, one, they can help control vessel diameter, and two, they prevent platelets from sticking to the walls, which could eventually occlude blood flow if that did happen. Okay, but every now and then, like I said, you're going to cut yourself, you're going to damage a vessel, and you're going to start leaking blood, and we have to stop that process. Now, let's say right here, we damaged a chunk of vessel right there. Okay, I don't know, let's say let's say you were doing a little bit of carpentry work, and a sliver went through your finger, and plunk, you pulled it out, and you started bleeding a little bit. All right, so now what's going to happen is within this kind of middle layer of the blood vessel, there's also going to be protein fibers, elastic fibers. There's going to be some collagen in there. Okay, remember collagen is a structural protein, which makes, any structure that has collagen in it rigid and tough. Elastic fibers, so the vessel can be a little more elastic and rubbery. So what'll happen then is when we break the vessel, we're going to obviously potentially damage some of the endothelial cells, so you won't have prostacyclins in this little area of the break. And two, what's going to happen is now you're going to have collagen fibers are now exposed to the lumen of the vessel. Okay, collagen fibers are now exposed to the lumen of the vessel. All right, so now you have something for the platelets to stick to. All right, you have something for the platelets to stick to. All right, but before that happens, before the platelets start aggregating together at the area of the broken vessel, what the vessel will do is it'll actually constrict. Okay, the physical trauma from the vessel alone can be enough to induce constriction. Okay, and that's important because think about it. If we narrow the lumen, if we narrow the opening of the vessel, and we have less blood flowing through the vessel, that means that's less blood we can potentially lose through bleeding. Okay, that's less blood that we can lose through bleeding. Okay, that, and also as we constrict the vessel, these exposed collagen fibers are now farther into the lumen, and it's easier for platelets to also stick to them. All right, so like I said, one, the physical trauma makes the vessels constrict, and also the remaining endothelial cells can also again release vasoactive chemicals to make stimulate the vessel to constrict. Okay, we would say that's the first stage of hemostasis, which we will call, we'll break this up into 3 stages. The first stage would be vascular spasm. Okay, just the vessel constricting whenever there's physical trauma to it, whenever there's a break in it. Okay, vascular spasm. Okay, the second stage, okay, after the spasm will take place, is what's going to be called the platelet plug formation. You're going to form what's called a platelet plug. Basically, it's pretty simple. Basically, what's going to happen are platelets are going to start sticking to the exposed collagen fibers. Okay, platelets are going to start sticking here. Okay. Now, what's going to happen as these platelets start sticking here, you're going to start to see a positive feedback cycle. Okay, you're going to start seeing positive feedback. All right. Now, what's going to happen is as these platelets start to stick to the exposed collagen fibers, they're going to degranulate. Remember, these have little granules as well, and they're going to release chemicals out into the lumen of the vessel. Okay, and the major one they're going to release is called ADP. ADP. Okay. Now, what happens is ADP, what it does is it's an attractive chemical. Now, we say this is positive feedback. Remember, with positive feedback, that's when we have some kind of physiologic response, and the response continues to get greater and greater and greater and more amplified over time. Okay, so what's going to happen here are the first few platelets are going to stick to these collagen fibers, they'll degranulate, release ADP, and that'll attract more platelets to stick to this area. And as more platelets stick to the area, that's going to mean more degranulation. And as more platelets degranulate, more and more and more platelets are just going to continue to aggregate here. Okay, and they're also going to release a chemical called thromboxane A2, and thromboxane A2, basically what that does is, and they're going to release serotonin. Sorry, I forgot that. Serotonin. Thromboxane A2 and serotonin are basically going to promote the maintenance of the vasospasm. Okay, so the trauma at first, when the physical trauma of the vessel will induce the spasm, and then these two chemicals will continue to maintain it. They'll continue to maintain muscle tension and keep the vessel constricted. Okay, and then more and more ADP is going to get dumped out, more and more platelets are going to stick to the area, and eventually what's going to happen is you're going to start to plug the hole. That's why we call it a platelet plug, okay, which is a good thing. But the thing is, though, is this platelet plug isn't going to stay together on its own for very long. Okay, it's not, the platelets are sticky, and as they aggregate together, they will stick together, but the problem is they can't stay together very well on their own. And that's where the final stage of hemostasis comes in, that's called coagulation. Okay, coagulation is the actual formation of a clot. Okay, now, what's going to happen here during coagulation is we are going to convert the plasma protein fibrinogen to fibrin. Fibrinogen to fibrin. Okay, so you see that? Okay, fibrinogen to fibrin. Okay, now basically we need to do this, like I said, because the platelet plug can't hold on its own for very long. Okay, so now basically you have these little proteins kind of circulating around your blood. They're called clotting factors. Okay, clotting factors. And these clotting factors, these remain inactivated. Okay, these remain inactivated unless we break a hole in a vessel and we start releasing these ADP and all these other chemicals, and then they'll be turned on. Okay, then they'll be turned on. And basically, there are two major pathways in this process. I don't really feel the need to go into this because it's just for what you guys have to do. It's just not exactly relevant to go over this step by step because it's a very complex pathway. But the end result of clotting factors is to convert this plasma protein fibrinogen into fibrin. Okay, that's the point of this. Now there are two pathways of this: an extrinsic, an intrinsic pathway involved in clotting factors. Intrinsic means occurring within the vessel, the area of the break. Extrinsic, kind of outside, in the lumen. Okay, but the end result is the conversion of fibrinogen to fibrin. Now what fibrin will do is it'll form a mesh-like network. Okay, it'll form this protein mesh-like network around the clot. Okay, and what that'll do is it'll secure the platelet plug, and it'll help to hold it in place. And then once we fully form this clot and completely plug this hole, and then the bleeding will eventually stop, and then we can start to undergo the repair process, the tissue repair. All right, but like I said, in a nutshell, that's kind of how this hemostasis process works. Again, when there's physical trauma to a vessel, vascular spasm. Okay, the physical trauma will make the vessel constrict, and then what will happen, our platelets will start to stick and aggregate to these exposed collagen fibers, and then that's what we call the platelet plug formation. And while we're undergoing this platelet plug, this positive feedback response, these platelets will start to dump out ADP, which then attracts more platelets to the area, and that'll make more of them degranulate as they stick there. And they'll also release thromboxane A2 and serotonin, which again helps to promote the vascular spasm and makes the platelets stick more tightly to the area. Okay, and then last but not least, we activate clotting factors, and clotting factors basically allow us to form a blood clot, which we call coagulation. And again, the real point of this is to convert the plasma protein fibrinogen into fibrin. And then once we form this mesh-like protein, this fiber network around the platelet plug, then we secure the clot. Then we can better control bleeding and the process which we call hemostasis. Okay, now the one thing with clotting factors is that there are some people out there that are born without clotting factors, and then they're more susceptible to bleeding. Okay, because if we take away any one of these clotting factors, there's many 13 to 20 different proteins that are involved in this process in a conversion pathway that we use to make the actual blood clot. If we're missing any one of those, that could cause a person to have a bleeding disorder, hemophilia, for example. So as a result, again, that's a genetic disorder, and that's just something you have to clinically treat, depending on the situation. But that's really platelets in a nutshell. That's hemostasis. Hope this helps, and enjoy the rest of your weekend.